right. So thank you, everyone, for joining us, SynBioUK, today for our first workshop of the series, Fundamental Tools for Synthetic Biology. For those of you who may not be familiar with SynBioUK, we are a student-led society and UK-wide network or a community that has its mission to connect academia, startups, and students passionate for synthetic biology. And as you may be familiar or not with, from experience even possibly, there is an increasing focus on the potential real-world applications of synthetic biology. And so we think, and hopefully you agree as well, that there is importance in equipping future scientists and synthetic biologists with the skills needed to bring your ideas to life and contribute significantly to the field. So that is really the main goal of this workshop series in general. And with this workshop in particular as well, we want to introduce you to a range of resources and tools to build fundamental skills in synthetic biology to then apply to your own projects. This workshop in particular, will focus on how you can use biopart databases. So for example, iGEM registry, which some of you may be familiar with, AdGene for plasmid backbones as well. So you can understand the main elements and considerations in constructing your own plasmids and transcriptional units, and then select these according to your experimental design. This workshop is going to be led by Davide Annese, who will also briefly introduce himself. But just right before I hand it to you, Davide, I want to encourage all of you to participate in following along with Davide's demonstration through these databases, asking questions in the chat area throughout the demonstration. And at the end, we also have a Q&A section just devoted to this, to asking the questions that you want. So if for some reason you do encounter any technical difficulties in following these databases that Davide will be demonstrating, feel free to message me privately in the chat as well. I also invite you to attend future sessions, future workshops. SynBioUK, this is for now a 3-part series workshop until Christmas. And these are 2 next workshops then, next week, Wednesday, and the other week, Wednesday as well. And please engage with the SynBioUK community as well through social media, Slack, and at the end of this workshop, specifically to give feedback on this workshop, you will be directed to a feedback form, and you also have details of where to contact us and engage with the SynBioUK community. So that's it for me from now. I'll hand it over to Davide to introduce himself and to kick off this first part of the workshop.
Okay, thank you for the introduction. Hi, everybody, I'm Davide. I work at the moment, so my background is in biology, and I did a master in Edinburgh in synthetic biology, and I graduated in 2021. So it was during the pandemic. It was kind of a nightmare, but that everybody has gone through it. So afterwards, I found a job as a research assistant. So I'm not a PhD student, but I'm working kind of, it's a similar position, but you don't have the recognition. In the laboratory of Jim Hasloff in University of Cambridge as a synthetic biologist in plants, and I work with a small bryophyte called Marcantia polymorpha, and I do all sorts of stuff with it, and yeah, that's another topic. So for today project, we're going to introduce you to the construction of a plasmid. We have got a prompt that was provided. Let's see if I can share. Okay, this should be fine. So for our today's project, we're going to build a plasmid with the objective to constitutively express GFP at a high transcriptional rate, but at a low translational level in a kanamycin-resistant backbone in Escherichia coli. This has to be done with directional cloning, and the enzymes that we're going to use are EcoRE and PSCI, which are the 2 of the 4 enzymes involved in BioBricks, which is the standard for iGEM, or used to be the standard for iGEM at least. So to achieve this objective we're gonna need a backbone, obviously, which is gonna entail the kanamycin resistance and the origin of replication, which controls the number of copies that is usually inside the bacteria. We're gonna need a constitutive promoter because that's the objective. So we want to constitutively express our protein. We're gonna need an RBS, which is a ribosomal binding site, and it's the point in which the ribosome binds to the mRNA and start the translation into protein. So it starts the amino acidic chain. And then we're going to need GFP variants as a CDS part, which is the proper protein that is going to be expressed. At the end, we're going to need a terminator as well, which is the sequence of RNA that is going to tell the polymerase to stop transcribing. So all of these, where can we find these parts? There are 3 main repositories at the moment, especially for whoever is keen on joining iGEM or similar project like IDEC or whatever. One is the iGEM repository, which is the most famous. The NCBI repository is the one that is a bit more professional, and it's kept by some international institutes, and is the one that researchers mostly use. And then we have SynBioUK, which is a new hub that has been developed here in Cambridge, actually, from Jenny Meloy's lab, and its aim is to kind of tidy up all the different repository and parts that are online that you can find around. So to start everything, we start with looking at for a constitutive promoter first. So we go on Safari. We look for our iGEM registry. So go on Google and Google iGEM repository. And the first result is usually this page. We can do it together if you want. Stop. iGEM repository, the first result is iGEM parts that you open up, and you're going to find yourself in this page. So the iGEM web page is a little bit slow in loading, especially if you're working from a low Wi-Fi connection. So watch out for that. But beside that, what we're going to do is we're going to look for parts. So there are some very interesting tools that it proposes you for learning how to assemble, what to look for. There are the websites, the old websites, there are the wiki for all the every year, every group has to build their own website, which is the wiki. What we are mostly interested in is going to the catalog, which is the one that contains all the parts. So it's the repository, the real proper repository. We're going to do browse by type, and we're going to look for promoters. We type in here, we go to this page. If you go to promoter design, there are some helpful resources that teach you how to do it. They also teach you kind of what the BioBrick system is. And then if you go to promoter help, you're going to find mostly also other tutorials that are more basic. We're going to go to promoter catalog. We open up and we find ourselves, the catalog is divided in this section. Some is cell signaling, constitutive promoters, metal sensitive promoters, phage promoters, and so on. What we're most interested in this case is constitutive promoters, so we're going to go to this page. Bacterial promoters are usually very short, so this is a good thing for when you do the assembly, because the longer the parts, the harder it is to assemble, because they form secondary structure and there is to be, it diminishes the probability of the sticky hands to find each other. Okay, so this is a page of the constitutive promoters. There are different promoters that are recognized by different sigma factors, which are the transcription factor in Escherichia coli, sorry. The most used one is sigma 70. Sigma 70 is the one that is most widespread. It has expression along all the cycle, especially during exponential growth. but still also during the stationary phase. If you're more interested, there are different types. If you're more interested in something that produces only a stationary phase, so you don't want to produce it during the exponential phase because you want your bacteria to grow faster or more easily without stress, then you can try to go to this order, sigma factor. You should go through each of them and decide which one more interest your objective in general. Okay, so once you arrive to this page, you start looking at the promoters and you try to understand what you're looking for. So we're looking for a promoter that is constitutive and it has to be quite strong. We want to have a high transcriptional rate. So what we look for first, we start opening the list that we have because we don't have many other page. You will see that most of them will have this kind of page, which is not really descriptive and there's no quantitative data. It's usually not very useful. It's a little bit of a leap of faith to use these promoters. They need to be characterized every time, so it's not the best way to choose a promoter. If you scroll down the first one, I really look at them, so you can look at them with your time. They're usually all kind of similar. The one that we most interested in are going to be the constitutive promoter family member, which are from J2300 to J2318 or 19. The 19 is the wild type, and all these other are have been developed, so they are synthetically made. We should try to open the first one, J2300. And we are arrived to the page of the promoter. So in this promoter list, we've got a list of several promoters. They're all related to 112, which is the weakest one in this list. It's considered to be the value 1. Everything else is compared to it. So how many fold times is stronger the signal than the thing. This is a signal that is recorded by a plate reader. So what you're looking at is the protein production and not the mRNA production. So since our intention is to have something that is transcribes at a high rate, we're going to look for qPCR data. What you can do is to go look through all these pages. There's a lot about it. So you can find a lot of experiments, many groups worked with them. So we like this page, we like this kind of promoters. So this is what we're looking for. We're looking for something that has pages that are very comprehensive with different experiments used by various groups, and they usually have quantitative data. This family is one of the first ones that they were developed, so we have a lot of data about them. So first of everything, we're going to look for which one of these has the highest mRNA expression. So what we can do is we can look for qPCR directly, so we don't waste time. We arrived to Argentina, Stuttgart in 2019. You can go through what they say, but they eventually test some of them, 100, 112, and 118. So 100 is the one that has the highest expression, while 118 is actually not very well expressed. Even so, when they do the qPCR, which tests a different time, they realize that the promoter 118 is still transcription way higher than 100, which is the one that is mostly used by IgM teams and is the strongest for the expression. There might be different explanation for these, but we're not very interested in that. We just know that promoter 118 is very strongly expressed, very strongly transcribed, even though the expression is not reflected in it. So what we do is that we can go back. We're going to go to 118, which is him. We open the page. We're back. It's the same page because mostly they often have common experiments. So you will see pages being a little repeated sometimes. In each of page, you will find this small square here where you can see the Build-A-Plasmid or get part sequence. What we do is that we get part sequence, we copy, and then we go to, in this case, Benchling. Benchling is a platform that we use for gene engineering manipulation in silico, obviously, so in a laptop. There are different other software. Benchling is an online platform and is shareable, which is something that usually teams inside research groups really appreciate. So we put it here. You can also use other software. For instance, SnapGene is very famous for what we do. We can initiative, make an annotation, and say this is our promoter. Okay. Okay. This is going to be important in a second later. Okay, we go back to our parts. So we found a promoter that is highly transcribed, even though it's not as highly translated in C. And so you can see that 118 is actually not as strong as 100, but still quite a strong promoter. You can see it's 1,400 times stronger than the baseline that they use, which is one of the other promoters. Therefore, we decide to use this one. We go then for an RBS. We go back and we look for an RBS. So RBS are the, we follow the same path. So we go back to catalog, RBS. We arrive to this page. We go again to the catalog. Here again, there are tools that you can use and follow to understand better what the RBS is, what it does, and why we need one in bacteria. We go to the catalog. There are two main families into the iGEM repository, plus some more that have been annotated that you can find following the link here. What we are most interested in this case are Anderson RBS library or community collection. Community collection has 8 of them. All of them are kind of well characterized. They've been used a lot by the iGEM students. So we're just going to go for RBS in the library, Anderson library. Okay. This is the description, what's inside, what's not. This is how the structure of the biobrick is made. So there is the biobrick prefix that has the enzymes inside. There's a XBA1 enzyme in between, and they're usually as a part, they are sold. They are sent by iGEM with the promoter J23100, which I've been seeing is the same family of the promoter we chose. But in this case, we're not interested in this one. So we go through with them, we look at what we decide to do. There is a list. If you run down them, so they haven't been characterized singularly because it's quite difficult and we're not used to characterize RBS usually. So we mostly, they have been characterized in a library that was much larger, it was more than 150. They published this picture, which is not very representative. It's actually quite bad quality and not very useful. But at the end of the description, in the measure strength, they say that, they are all, they transcribe all at a rate that is above background, and they're very roughly in decreasing order of activity. So in this case, RBS controls the translation, and as we looked online, we wanted to find something that has a lower translation rate than a transcription rate. So instead of selecting one of the top ones, which are going to have a very strong translation, we can check for something that is a little bit weaker. We can maybe go for, I don't know, we can choose the 134, maybe this one. Yeah, we can open the page. The library that encompasses all of them. We go again to this small box here, get part sequence. We copy the sequence, which is very small. It's just a ribosome binding site containing a shiny cardinal. We go to benchling. We add it after the our promoter, and we call it RBS. Create annotation. RBS, and the part is J. I don't know. It's 61134. 61134. And we've done this. Okay. And so doing this every time, we are gonna copy and paste all of these parts one after the other inside your sequence handling software of your preference. In this case, it's benchling. And then you're gonna arrive to a point in which you're gonna have well, I'll show you later. Well, let's look for the other part that we decide to look for, which are after the RBS is gonna be the EGFP. For this case, we're gonna use SynBioUK. SynBioUK is the newest hub that we talked about, that I told you about. We can use it for looking for a GFP. So in this case, since we are expressing something at a low translation level, we are going to look for something that is a little bit stronger than wild type, than the wild type GFP. So we're going to look for an enhanced GFP version. Enhanced GFP is just a variant that is more commonly used. Why don't you find it? More commonly used and I don't know why SynBioUK is not working. So SynBioUK tends to be a little bit slower, so watch out when you use this one. Okay, we found. So we look for enhanced GFP. The first GFP defines are not exactly green fluorescent proteins, so they are still variant and they are enhanced, and they are called YFP, CFP. This is yellow fluorescent protein, cyan fluorescent protein, and so on. So we go through them quickly until we find a GFP, which is here. It's the 8th result. We open the page, we open the link, following this SynBioUK, which is very slow, I'm sorry. Okay, we arrive here. The first thing that we notice is that it says that this part has been discontinued, and that this part was inside it was already inside it wasn't alone just the AGFP, but there were also some flanking regions containing the terminator. So we follow what they say. Please consider using BBA SO3452. So we go for this part. I think can very slow, sorry. Okay, we're here. So we can probably find the sequence visualization. We can find the sequence directly here if we want to, but it's messy to copy because it's not a FASTA format. So what we do is we go directly to the source, which is the iGEM registry again. So this is the page where you find the GFP, the enhanced GFP that was used by the for the first time by the iGEM team. This is the excitation emission peaks, which are kind of close. The first in intensity, and if you scroll here, you can find the sequence. So we can do get part sequence again. This is the whole sequence. You can see that it starts with ATG and it finishes with TAA. ATG is the first codon, is the methionine, and the TAA is the stop codon, so it transcribes for nothing. What we do is that we copy this all, we go to our bench link, copy here, back, annotation, and we call it copy the sequence name just in case we forget what it is. Okay, save. Now we're gonna have to look for a terminator. And so first of all, guys, if you need me to go slower or if you have questions while we talk about the iGEM registry or the various registry where we're using, like the Sci-Hub, the SynBioUK, just let me know. You can stop at any moment. I know it's a lot of information all at once, so feel free to stop me in any moment and ask me more interesting questions or whatever you need to, if I have to slow down or whatever. Okay, so we found the sequence. This one we don't serve any use anymore. Okay, now we're looking for a terminator. We should go back to the iGEM registry again. So we go repository, sorry. We go back to the repository. We follow the same path we used before. So we do catalog, browse by type, and we see, come on. And we found terminators. So we open our terminator page. Again, useful resources if you need to learn what they are. There are two types of terminators in in general in bacteria, especially in Escherichia coli. One is autonomous, so the DNA, while the strand of mRNA is synthesized, it forms this little hoop, this little pin that interacts with the, I think it's the C terminal, if I'm not wrong, of the polymerase, and stops the polymerase and give a sign or something happen by which the polymerase stops transcribing and detaches from the DNA. Or there is a raw activated and raw controlled. So there is this protein that usually finds the mRNA and scroll through the RNA until it arrives to a polymerase, and then stop the polymerase when it finds a certain sequence as well. The certain sequence is usually a sequence that has that slow down the polymerase, so the raw protein has time to catch up and stop the protein for transcribing. Even here, there are different terminators. There are bidirectional terminators, which are terminators that are capable to stop the polymerase coming from both sides. And there are type of terminators that are only capable to stop the reverse strand of the polymerase. And then there are eukaryotic and yeast, which we're not going to use today because obviously we're working with prokaryotes. The most commonly used ones are forward terminators. So we're going to open this one. It clicks us down just immediately here. So we can follow the first one. You can see that the first one are coliphage terminator. For instance, T7 is, I'm not sure about T1, but T7 is also phage origin. T7 is originated by the phage T7, which is a phage that infects Escherichia coli. We are not using a promoter that uses it, and we don't know if we're going to use lines of Escherichia coli that contains the polymerase that can recognize the T7 promoter and terminator. So we exclude all of these all at once. Very immediately, the fourth result is double terminator. It's a composition of two different terminators as well. We open this one. We can read around and decide what to do with this one. The fact is that eventually having a double terminator is good because the terminator is capable to, more terminator in tandem are capable to increase the termination efficiency and therefore increase the efficiency by which the terminator is capable to terminate the transcription. I don't exactly know about how that works in bacteria because I work with plants, but what happens is that the less the polymerase stays attached to it, the easier it is to splice the RNA, or no, well, not splice in bacteria, but to further work with the mRNA. So it usually increases the translation rate as well. So we go to the get part sequence. We copy this one. We go back to our expression cassette. And we paste it. And then we just call it. We call it we can call it okay. So our construct is almost ready. We have a promoter, we have an RBS, and we have the CDS, so our enhanced GFP. We have our terminator, which is going to stop the terminate transcription. Actually, I might have made a mistake because I'm not sure this one is going to work because it's for T7 as well. But well, anyway, you have a long list to choose from, so you can look for the ones that you prefer. Just make sure that they work with normal polymerase. They don't need the T7 polymerase. Beside that, we can go back to our main project. So we have all the parts for making our cassette, the expression cassette. We have a promoter, RBS, a CDS, and terminator. What we need now is backbone in which to insert them. And to find a suitable backbone, we're going to look into AdGene. AdGene is a repository that now that usually from which you can buy all the backbones or plasmids that you need. They have an infinite list of amounts of different ones, so it can be a little bit overwhelming and it could be a little bit difficult to find. We can try to look. I will let you do it. I will let you try to look for a backbone in this case. I'm not gonna show you everything. What I'm gonna show you is how you can use Adgin. So the first page of Adgin is this one. Adgin, we're here. This is the homepage of Adgin once you open it from Google. What you can do is that you're gonna look for something that has Echory and the other one, PST1, and then those two enzymes are inside the BioBrick syntax. So they're using BioBrick. What I would advise you to do is, since you can't filter for what enzymes you want to have in those plasmids, what you can do is that that they are going to be in the BioBrick, so you can look for a BioBrick component, and then we want them to be in kanamycin, so we can also look for something that is in kanamycin. BioBrick, we say kanamycin resistance. It's just 2 keywords. You should be able to find them. We can look directly in plasmids. So if you don't look in plasmids, you're gonna have to choose afterwards. It's gonna tell you catalog in plasmids or educational resources. Educational resources are useful tools to learn how to do these kind of things. So we go to plasmid, and then we've got a long list of plasmid that we can choose from. You can filter so that you can reduce your result, because you don't want to go through 111 different backbones to decide which one is the best one for you. We know that we want to bacterial expression, so we do yes, bacterial expression. Okay, and we go about 74 variation. Synthetic biology, I never selected, but you can if you want to. We usually look for. Okay, so we can do species Escherichia coli. So we know it's going to be in Escherichia coli. Yeah, you're going to have to play a little bit with the filter because some exclude each other and it's a little bit annoying. So we want something that is in kanamycin. Resistance marker. Okay, so we can do more filters. We can do resistant the kanamycin. So we have 7 results at the moment. P-cons RFP, P-cons RFP, P-cons RFP, P-cons RFP. These are all similar primers from the same library. Okay, so I'll let you play with the filters and play with the BioBrick with the keywords. You can also look for just more specific naming if you know already what to look for. And then I would say maybe 5 minutes and whatever you can find. Okay, I see there is a question. So maybe I'll answer the question in the meantime. People can try to look for themselves some Adgin. Once you find the gene, once you find a plasmid that you think is suitable for what we're looking for, you can paste the name in the chat and we can go through them and see if they're suitable for what we have to do or not. So as for the question asked from Teresa, so she asked, so we only need to paste each part directly to benching and the program assembles each part in order. So kinda. What you can do is you go to the part, you go to on benching, for instance, and I think Snapgene is gonna work the same way. You can directly paste a sequence into. You can upload a sequence directly. If you, for instance, from Adgin, we can download it. I'll show you as soon as we have one. Download one from Adgin. We're going to import DNA and import it, and it's already all annotated, so you don't have to do anything. I didn't recognize all of the parts inside the plasmids because they're annotated from Adgin indeed. Then you're gonna have to assemble, but I'll show you later. In this case, you copy and paste the sequences one after the other, and you can also copy and paste sequences in the middle of the others. So like here we can do insert bases and write, I don't know, ATT, GCT. this is my cup side, so you can recognize it. You can see it here. Obviously, if you do something like that, you're gonna break your frame, or you're gonna add amino acids that you don't want. So just don't do it because it's not really helpful. But you can also add sequences in each point of the benching sequence you have. For in this case, I was adding one after the other because we were going in order from promoter to RBS to CDS to terminator. But for instance, if we find a GFP first, we could have added the promoter afterwards by going at the beginning and doing its bases, and then you can like paste them here. As for the assembly, I'm gonna show you as a last thing once we have everything. You can do create new assembly and look for digestion ligate, gypsum, or golden gate, which are different types of assembly method. Okay, let me know if someone finds any plasmid or any plasmid that satisfies our needs. So there is a new question. Where would you look for information on which polymerase a strain a microorganism has if you haven't worked with it before? This is in relation to find the correct terminator sequence. Okay, so I guess we're talking about Escherichia coli in this case, which is these most widespread organisms for bacterial and synthetic biology. What we do is, I don't know, let's say that we know already what strains we were working with. So strain of being organisms. So usually they have their native polymerase as usual. So each organism has its own native polymerase. And so when you look for a promoter, you have to look for promoter that are recognized by the specific organism because it has its native polymerase. In the case of having, so for instance, before when I was talking about the T7, is that there are polymerases that are transcribed by phages, so they are transcribed by the virus that infect the bacteria. And when the virus inserts the piece of DNA inside the bacteria recognize the promoter on the virus strand, transcribe the polymerase from the bacteria, from the virus, sorry. And that polymerase, which is called T7 polymerase, is going to recognize only promoter for the T7 polymerase, and is going to be stopped by only terminator for the T7 polymerase. This is just a technique that is in biology. What we did is that we harnessed this with polymerase. For instance, some engineered strains of coli, which, for instance, BL21, I think, has T7. I'm not super sure. For instance, in this case, we want to know, we go to the page that sells it. This is the Thermo Fisher page. It says BLT1 DE3 competent cells, which are cells ready to be transformed. You go to the transcription, contains lambda DE3 prophage that carries the gene T7 RNA polymerase under control of lucky UV5 promoter. So it already tells you, usually the page tells what's inside. So if we look for another one like the H5 alpha, which I think does not have it, but I'm not 100% sure. No, it doesn't say. So this one probably does not have it. There are also, every time genotype is all the mutations that are inside this specific strain and why. Okay, has anybody found any plasmid that they think are suitable backbones? Okay, so if you look what I've been doing, I've been looking for a BioBrick standards. So I just look for BioBrick. These are usually contains the BioBrick CSR, which is a multi-site with all the enzymes involved in the BioBrick and some others more that you can open and use as what you want. Each plasmid has, so these are all single guide RNA. So these ones are for CRISPR-Cas9. We're not interested. PTJ. Let's see if you can find. Okay, let's say that we look for this one. We open it. There are a lot of information. What we are looking for is, okay, so the copy number in this case is low. Low copy number means that there's going to be less copies of these plasmids inside the bacteria because the ORE, which is the original of replication, is not very strong. So the plasmid is kept at lower concentration. So if you're looking for a plasmid to do a mini prep where you want to do cloning, so you want to amplify your construct, this is probably not the best one because it has a low copy number, and you want to have a high yield when you do this kind of experiments. More is in ampicillin, so this is not the backbone we were looking for. We can exclude it. We can go up. EDL3, ampicillin again, low copy again. We don't like it. Are they gonna be all the same? It's on the copy. Spicy. 11. So I changed a little bit the filters and I changed the keywords because I want to have something kanamycin and with the BioBrick. So this is an empty promoter, BioBrick. It's in kanamycin. Growth strain is the H5 alpha, which is the strain we was talking before. It's in high copy. So this one could be a potential. What we do when we find something that matches what we need is that we go to see the sequence, view all sequences. At this point, it gives you the FASTA format where you can copy and paste. It gives the GenBank or SnapGen format. It depends which, if you're using SnapGen, for instance, the software, obviously you download SnapGen. For benching, we're going to use GenBank. Anyway, let's see our first side. We can open an analyze sequence and see, we immediately see there is a echo RE here, RI here, and there's going to be PST, we can find it. PSTR either here. So this promoter, this is probably not great because it has a PSTI and echo RI into the one is in the RBS, like close to the RBS, and the other one is in the middle of the replication with the ORI T7 terminator. So we probably don't want to touch this too much. It's probably not the best. You can go back. Actually, kind of lost. So the flare here is the option that tells you how many people is requesting it. So how much is it's an indicator of how good something is usually in AdGen, how reliable it is. Okay, so I can't find the promoter I've seen before, but we can go back. It's like the time is running out, so I don't have time to go and look again why I can't find it. But I've looked it free before and I found one that was kind of suitable. I downloaded the GenBank and I've got it here, so we can go. I'll show you how to do it on benching. We do add DNA sequence, import DNA, and we can just drag and drop. Okay, so it has uploaded the gene. This is the whole plasmid, as you can see. Has we don't really need this one. Okay, it has a laxid, and it has ampicillin and kanamycin resistance. So we're most interested in kanamycin. Ampicillin is just a secondary effect. We don't really care too much. It's a high copy number. I know because I remember when I looked for it in the website. But anyway, so what we do now here is going to be looking for ECORE and PSTI. ECORE and ECORE and ECORE, and the other one is PSTE. Okay. So we look for them. Where are they? Can go to where they are. No. Okay. Okay. So you can see in this case they are in the MSC, which is the multi-cut site. It's a site where, as you can see, there are like a lot of different promoters. This is because this is inside the sequence of lacazid. Lacazid is a reporter. So if you transform this plasmid into a bacteria without adding anything into it, this bacteria is going to produce beta-lactosidase. Yeah, I think so. And it's going to digest the xgal, which is sort of sugar, a type of sugar that we add to the media, and it's gonna create a color which is blue. So you're gonna see that you're going to see that the colonies are blue, so they're wrong. At this point, we use there are the two PSCI and ECORE. We want, according to where we copy the ECORE sequence. Copy, and we add it at the beginning of our cassette, which we called expression cassette 2. So this is our cassette. We add it at the beginning, and then we do the same for the other. So we do we copy the PSTI as well, PSTI. Sequence, and we go to expression cassette. We copy at the end. Okay, so we have our sites here. We have our sites and we have our backbone, and the expression cassette is complete. What we can do now is that we can digest and ligate the product. So we see that go inserted. So as you can see, what I've done is that I added the ECORE as initial and the PSTI as ending, which is a little bit wrong. It's the contrary of what the plasmid is. So this could be risky. What I used to do is to just copy the whole sequence, do copy, and copy the reverse complement of it. So it copies the negative strand, and then what I do is I just do this way. So you copy and paste in the whole sequence, and it converts it completely. So now we have takes a second, but there's also annotation on it. The PSTI is at the beginning, but it's actually the ending of it because you can see the terminator is there, and the beginning is at the end this time. This was just to because benching is it doesn't recognize if something is inverted or non-inverted, so you have to do it manually. There are other software I'm pretty sure we can do it manually. So what we can do now is that we go in the middle of the plasmid, always because the problem is that benching does not recognize if you tell him to do. So what we're going to do is to create an assembly. We do digest and ligate. We start, and it's going to ask you to add a backbone. And you can only do in this case by selecting. But the fact is that according to the index point, it's just going to select you in between the 2 points, and I can't select all the others. So it's going to try to assemble this small part with our insertion, which is not what we want. So what we do is that we re-index, which means that it considered this point a new 0 point. So we can select ECORE this time, and we can select the PSTI, which is now at the end. And we can do set from selection. So it automatically recognizes the sticky ends they ends up having. And then we do insert, expression cassette, and we do the same. We select equary and PSTI, and we tell him select from selection. It's going to tell you there's no mistakes. The two frames, the two sticky ends coincide perfectly. So we say yes. We call it a different way. We give it a new name, so new construct. Usually when you do things, you don't call them new construct, but you call them with the name of the promoter and the name of the CES. Plus, sometimes it depends how you are organized in the lab. You can add the backbone you're using. Okay, so this is our final product. You can see plasmid in here. Oh, the annotation got screwed, but it's fine. Just don't care this. We can see our insertion has been copied fine. So this is our GFP. This is our promoter and RBS. You can see them. And this is the ending product. What you can also see is that the insertion was in the middle of the laxid protein. So the laxid protein has been opened up, and then something else has been put in between that has its own promoter and terminator. So laxid is not being transcribed anymore, and the colonies that have these plasmid are going to be white instead of blue. So you can select which colonies are the good ones or not after you do the transformation. This is the ending of our workshop, and I'm open to questions. I'm sorry I'm a little bit late.
Yeah, thank you so much, Davide, for that part of the workshop. While people actually come up with their own questions, and of course, questions can range from things specific to the workshop to things that you are particularly interested in that you would like to know if you can use these databases to apply to your own research and to your own interests. So just ask around, ask anything, and then whatever can be answered, I'm sure David will do his best to do so. I actually had a question myself related to the registries.
Mm-hmm.
And the example that you gave was on E. coli expression. You work with plants, and I'm sure people in the audience would also like to know how or whether there are parts available on the iGEM registry that could be used for plant expression or in mammalian cells as well.
Yeah, so there's some in the registry. Truth is that since the iGEM competition is in a short period of time, people don't usually work with plants, and they mostly work with bacteria. This is because working with plants is kind of long. Plants takes a long time to grow, takes a long time to be transformed and create generations and stuff. So when you do iGEM, you just don't have the material time to work with them. So there's many less on them. For this kind of experiment, you can mostly use the NCBI, which is the one that I said is a bit more, NCBI gene. This thing is behind my search. NCBI gene, which is another repository. The NCBI gene has more, all the genes in here, they are usually linked to a publication, scientific publication. So they are generally a little bit more reliable and better characterized than the genes and the things that you can find in the iGEM registry. What you can do is that you can look for GFP and then select, for instance, Homo sapiens, Mus musculus, and these are all GFP type, or no, these are transcription factors actually, because it's not recognizing it. But you can look for there are different types of repository, as you can see. Some of them are used for assembly, collection projects. Some of them are a collection of data sequencing or other types of data. It's a very complex and very long to explain type of repository. And that's why we're not really talking about it too much today. But you can look for plant. So Mercantia polymorpha is the one that I work with. Super rare is what that is. In this case, it's giving you the whole genome. You can find the genes in here. What you usually do is that you go look for the publication in a real-time context, in a real-life context. You're going to find a publication that you're interested in, find the gene that you're interested in, and then there are software online, there are tools online that are used for codon optimize. Also, when you're on AdGene, you can often buy whole clusters, a whole group of plasmids all together. And those plasmids contain a list of things that are usually really adapted for the plant or the organism you're working with. So that's how you mostly work with. And for the iGEM registry, the registry distributions. These are tools. Okay. I don't remember exactly where was it, but you can go to promoters, for instance, promoter catalog. I can't remember how to arrive to that point, but there is a section in the iGEM registry that is dedicated to mammalian cells and to eukaryotic parts. And in that case, is everything in here is already code optimized for eukaryotic cells like plants and animals.
All right, yeah, thank you. Hopefully this also been helpful for some of the people in the audience. I think we had another question here by Teresa Garcia. If there was any other recommendations for books or literature specifically that you could use to build plasmids?
I don't know exactly about a book. What I did when I was learning, because I also did iGEM when I was in my master, was to look a lot of YouTube videos. This is because the assembly system, what you do in a real context life, when you're doing real research, is that you usually adapt to what the laboratory already has. So they probably have already assembly system that they're using, which could be Golden Gate, could be Gypsum, could be BioBrick, or could be something else. The BioBrick assembly method is nowadays a little bit obsolete, let's say, because it's not very flexible, and it's very long to prepare, and the reaction takes many like the reaction are very long, and you have to purify the plasmids in between very often. What we usually mostly now is more assembly that of the type of Golden Gate. They use type 2S enzymes, which means that they recognize the sequence, but then they don't cut inside the sequence, they cut outside, just in front of it. And then you can change that 3-4 base pair that they cut with whatever syntax you want, and so you can stick them in 1,000 different ways, because you can change it as with any letter you want or any sequence you want. So there are assemblies that are automated, and they are really based on this suffix, so you can directly do things without having to change them every time or purify everything, because once you create sticky ends that are personalized, so the backbone can't reclose on itself. Yeah, like some system of these are jump system or loop system. It depends what you're working with. For organism, jump is for bacteria, loop or moclo are for plants, or for algae sometimes. For videos, yeah, I would just to watch a lot of videos on YouTube. Some of them are very well explained and very well. So look at the type of assembly you're going to use, and how that works, and then go to the original publication, which usually has a lot of information in it. I know it's a little bit more complicated, especially when you're not in the field and you're not working in it, because I remember when I was a student and reading for the first time about the jump system, and it took me several days, probably around a week, to understand how that was working. But then once you understand how the things work, and once you get a grip on reading research paper and things, it's much easier to understand what's happening and what are you doing, and everything is made for a reason. So it's much more flexible and fast and cheaper as well.
All right, thank you for that. I might wait, but we might wait another minute or so to see if anyone wants to gather their thoughts and ask something last minute in the chat. And this can be, of course, related to any of the databases you saw today, and even related to your own interests. All right. If there are no other questions, I will tie things up here. So thank you all for participating in today's workshop. Thank you, Davide. I hope this has been very useful as an introduction for you to these databases, for the participants for these databases, and that by understanding these, you could also apply them to your own research that you undertake in synthetic biology. Again, a huge thanks to you, Davide, as well, for sharing your knowledge and experience on the topic and for making the workshop so engaging. We have more engaging workshops coming up. The next one will be specifically on protein engineering tools, and the other one more deeply on benchling basics as well, as you got a little taster here today in part. So please, for you, the audience, let us know your experience in this workshop. As this meeting ends, you'll be directed to feedback form that you can share your thoughts with, and it's really just 1 minute of your time can help us improve massively these workshops and these initiatives. So just take that minute to please give us some feedback. So SynBioUK is also planning other initiatives, exciting ones next year especially. So sign up to our mailing list. You can also see us on Instagram, LinkedIn, join our Slack community, and of course, reach out to us via email. All of these links are in the feedback form that you'll be directed to as this meeting ends. So please feel free to check those out and engage with the broader SynBioUK community. So again, thank you, everybody. If there are no more very last minute questions, I'll end the meeting here. And we'll see you around for our next workshops.